Hi, welcome to the Mitosis Notes podcast lecture number 2. We're going to be talking about the process of mitosis and uncontrolled cell division today. Please take notes like you normally would in class. The answers will appear in red on the screen. So last time we talked about chromosomes and karyotypes. So now that we know what chromosomes are, we're going to go through the actual process. And mitosis has 4 phases. First is called prophase, second is called metaphase, the third phase is anaphase, and the fourth phase is called telophase, or telophase, however you want to say it. Now, once the DNA has been copied in interphase and everything else is ready to go, we're going to do the mitosis process. Now, mitosis is done by all of your somatic or body cells. And at the end of the mitosis process, we're going to create 2 cells that have identical copies of DNA. Now for the first step, or the first phase of mitosis, prophase, we're seeing here that the chromatin is shortening and thickening and condensing into structures called chromosomes. Specifically, you're seeing the sister chromatids here, so the original and the copy together. And you'll also notice that the nuclear membrane, it's the membrane that surrounds the nucleus, is disappearing, so we can move the chromosomes around the cell, because otherwise the membrane would block it. And we also see the formation of something called centrioles, which are these purple dots in the diagram, and spindle fibers, which are in yellow. And these act like pulleys and ropes to actually move the chromosomes around the cell. Okay, so if we take a look at some cells, onion cells undergoing mitosis, the cell that's being pointed to by the arrow in green is in prophase. So you can see there's thicker structures inside the nucleus that are not present in this cell with the red arrow, which is in interphase. You're just seeing the chromatin as the fuzzy substance in the nucleus. Now the second phase is called metaphase, and we're seeing here that those centrioles and spindle fibers are now moving. So what they've done is they've actually moved from the sides to the opposite ends of the cells called the poles. Those sister chromatids also lined up in the middle of the cell. And the last thing that happened was those spindle fibers, which are the ropes essentially, attached to the sister chromatids at one end, and the other end of the spindle fibers attached to the centrioles. So if we take a look at these onion cells undergoing mitosis, the cell that is in metaphase is being pointed to by the green arrow, and you can see that the chromosomes are lined up in the middle of the cell. Phase number 3 is a short phase called anaphase, and in this particular phase, the sister chromatids are pulled apart into the original chromosomes on one side and the copies on the other. So we basically separated them into 2 sets. So if we take a look at the cells here, the one being pointed to by the green arrow is an anaphase, and you can see the chromosomes look like they're kind of moving to the opposite sides, and that shadowy part in the middle is the spindle fibers. The fourth phase is called telophase. We're going to get rid of the centrioles and spindle fibers because we don't need them to move anything anymore. And what happens is you get membranes, nuclear membranes, forming around each of the sets of chromosomes. So you're going to see 2 nuclei in the cell here. So if we take a look at the onion cells undergoing mitosis, at the cell on the bottom, you're seeing the 2 nuclei in one cell. That's how you identify it as being in telophase. Now, once you have the 2 nuclei all set up, now we need to actually divide the cell into 2. And that's where cytokinesis comes in. This isn't actually mitosis, it's after it, but it's a very vital part because we need to distribute the cytoplasm to each of the cells. So you've seen here that the cell's kind of been pinched off into 2 cells. The chromosomes have also unwound back into chromatin. So we're basically resetting each of these new cells into the interphase again. So we have 2 cells, 2 identical copies of DNA, and now they're back in interphase, and they can continue doing normal cell things. Now sometimes this process goes awry. So normally cell division is controlled by these proteins called cyclins. Now, these proteins may be ignored by the cell if it has a mutation or has been damaged in any way. And what happens is the cell will kind of go fast forward into mitosis, and it'll divide and divide and divide and form a tumor, right? A mass of cells. Right? Now, sometimes the tumor can be malignant, which means that it can destroy other cells and move around to other parts of the body, and this is what we would know as cancer. Now, sometimes the tumors are benign, so they're completely harmless. It's just a mass of cells that really has no harm to the body. So here's some pictures of tumors there. Alright, and these tumors, or basically cancer or benign tumors, can be caused by a number of things. It could be mutation in the DNA, toxic chemicals, radiation like ultraviolet radiation, so people who tan a lot are at higher risk for cancer because of this. And we talked a little bit about that when we did mutations. Okay, so please come to class tomorrow with any questions that you have about the notes, and if you'd also be prepared for an open notebook quiz on the notes in class tomorrow. I thank you for listening, and have a good day.